Then a few years ago, one of my colleagues, we teach a genetics degree in Swansea, based out of the medical school, and one of my colleagues, Miranda Witten, she is an entomologist, really, so she's working with insects and also parasites which the insects carry and transmit to humans. She came to me with the idea that perhaps we could try and exploit bacteria that live inside the guts of these insects to produce information which might help to block transmission of parasites by the insects. And this all came from work, initially from work which a now retired professor from Swansea, Professor Norman Ratcliffe, initiated. So he's now living in Brazil, where this particular insect is a real threat to health. So colloquially, the insect is called the kissing bug, but it's not a very friendly insect. It comes along in the night and effectively sucks blood from a human, usually lands on the face of the sleeping individual. And after it's had its food, it deposits some of its feces and on the skin of the individual, and the individual rubs the wound, if you like, because there's inflammation there because of the insect bite, and rubs accidentally the feces into the wound, and the feces contain the parasite. And that's the way the parasite gets into the human bloodstream. So we started to work with this insect, and myself and my group with the bacteria that live in that insect, which are distantly related to the bacteria that I was talking about, which produce antibiotics. And we had essentially genetic tools with which we could try and manipulate these bacteria. So in that way, using that technology, we could effectively use these bacteria as a form of birth control and prevent the females producing viable eggs. So that's a way of population control of the insect, which would, if put into practice, would then reduce the rate of the infection of the parasite. And we've now gone on to translate this technology we had funding, from the Gates Foundation, with which we could translate this technology first into a hugely important agricultural crop pest, and also in the tsetse fly, which is a vector of the parasite which causes sleeping sickness in Africa, sub-Saharan Africa. Swansea University decided to patent that technology. Now, at the time, the head of, if you like, tech transfer in Swansea suggested to me that couldn't we do something even more impactful, which I thought at the time was a bit cheeky, because I thought what we'd done was also already quite important. But anyway, I was reading an edition of New Scientist on the train. The article was predicting technologies which might come about in the next 10 or so years. And one of these technologies was to use information similar to the information which we'd used to deliver to insects, to use that information, deliver it to tumours, and stop tumours growing. I then looked into what sort of bacteria could we use which actually will selectively colonize tumours in not healthy tissue. We then attracted some funding from Cancer Research UK to effectively put our ideas into practice, to try and put our ideas into practice. And lo and behold, we were able to demonstrate that similar to what we've done in insects, using bacteria that now target tumours, we could deliver the information and stop tumours growing. And because the bacteria only infect the tumour cells and not the healthy cells, we predicted there would be few, if any, side effects. So that the side effects from these traditional therapies are almost as bad as the cancer itself. We've taken this forward. We've had Welsh government funding to help with this, and also additional funding from Cancer Research UK. So we're at the stage of what are called preclinical trials with this approach, and the results are very encouraging.